Thank you so much. It is a privilege to share my research and information with you. This is an independent study that I conducted in the summer of 2018 on the food provisioning, prey composition, and nesting success of ospreys in Northwestern California. For my introduction, I would like to propose a question. What can ospreys inform us about prey species composition across spatial and temporal scales? And is there significance between nesting success and food provisioning rates? I hypothesize that ospreys are effective tools that can provide data to managers, such as environmental and fisheries managers, and that nesting success is a function of high food provisioning rates. For millennia, humans have been using birds to help us locate fish. In fact, birds can indicate productivity and have the potential to be indicators of fish availability, which is then correlated to fish abundance. The osprey has a diet almost exclusively of fish, has nearly a worldwide breeding distribution, and can be found in both fresh and saltwater environments. The objectives of this study were to report on the prey species composition of a coastal population of ospreys, and two, to measure the relationship between nesting success and food provisioning rates. This is a study area map of Northwestern California where I conducted my osprey observations. Nest A, B, and C were located in Humboldt County, while Nest D, E, and F were located in Del Norte County near the mouth of the Klamath River. Let's take a closer look at a main foraging site, the mouth of the Klamath River. The yellow highlighted area is the general vicinity in which I conducted my osprey observations of them foraging. And what makes this place so special and dynamic is that I've never seen so many ospreys converge and forage together in such a concentrated area. And I'm thrilled that there are many efforts to restore the Klamath River. From May 15th through August 15th, I spent a total of 265 hours observing nests and the main foraging site. As you can see there, I have my viewing scope, my binoculars, and my camera to ensure that I capture and record as many fish items and species as much as possible. I identified each fish to the lowest taxonomic level to the least family. I calculated nest success and productivity by the method of Steenhoff and Newton. I compared provisioning and other activities between the successful nest and the failed nest and calculated the standard deviation of this difference. And to substantiate my statistics, I conducted Pearson's chi-squared analysis with Yates continuity contingent. We're now in the results. What was one of the most surprising findings was this: that is non-native American shad, Alosa sapidissima, caught at the mouth of the Klamath River. What was surprising was that ospreys weren't catching just one or two, but as common prey. This table represents the total number of fish items and species witnessed and observed. Out of the 514 fish, 20% was non-native American shad. The expected salmon and trout at 25%. Pacific lamprey, nearly 10%. Other important species included jack smelt, sculpins, smelt, starry flounder, surf perch. Now, unknown constituted 22%, as some of the fish were half-eaten and unidentifiable, especially at the nests. Also, I experienced limitations with the capabilities of my camera. Let's take a look at how prey species composition was distributed on a spatial scale. On the x-axis, MK, the first column, represents the mouth of the Klamath River, followed by the successful nests, Nest A, B, and E, then the failed nests, C, D, and F. On the y-axis is the percent composition. Prey species composition exhibited variation across all locations, especially at the mouth of the Klamath River and the successful nests, in particular, Nest A and B, which are the second and third columns. It is important to note that the unknown prey items, represented in purple, was expressed throughout all locations. At the mouth of the Klamath River, the first column, prey species composition was dominated by American shad, represented in magenta, the salmonids in turquoise, Pacific lamprey in dark gray. Nest A and B, the second and third columns. Those nests were located near Humboldt Bay, and it is assumed that ospreys forage near their nest sites. Therefore, prey species composition was much different than the mouth of the Klamath River. At Nest A, the main diet, represented in light blue, was jack smelt, while at nest B, the main diet represented in green was surf perch. Both of these locations did express sculpin in orange and starry flounder in red, which is interesting because those are benthic species. Nest E was the only successful nest near the mouth of the Klamath River, but since most of the fish were half-eaten, it was difficult to get identification. Nest C, D, and F failed, and with a very limited amount of prey items being delivered, it was difficult to acquire and obtain proper identification. Nest A fletched 2, Nest B fletched 3, you have the whole family there, great to see. Nest E, the only successful nest near the mouth of the Klamath River with 1 offspring. 3 of the 6 nests fletched progeny, therefore nesting success was 50%. Productivity was 0.83 young per active nest, and I'll go over the significance of this value in the discussion. The overall bigger picture of ospreys investing time in provisioning and other activities looks like this. On the x-axis, we have the successful nests, nests B, A, and E, then the failed nests, C, D, and F. The y-axis is a percentage of time. Blue represents the amount of time ospreys invested in provisioning, while gray represents the amount of time invested in other activities. As you can see, the most successful nests, nests B and A, ospreys spent a considerable amount of time invested in provisioning, while at the other end of the spectrum, the failed nests, ospreys allocated the majority of time on other activities. Let's condense this overall bigger picture into finer resolution. On the x-axis, I compared provisioning and other activities between the successful nests represented in blue and the failed nests represented in gray. On the y-axis is a percentage of time. The standard deviation for the successful nests was 18.7, and for the failed nests, 10.3, as indicated with the error bars. What is evident here is that the error bars do not overlap, and because of this, further investigation was warranted, that there could be something significant here and it's not happening by chance. And to substantiate my statistics, it was crucial to create and develop this two-way contingency table, which tests the significance between my categorical data. The test statistics obtained here were the chi-square test statistic and the freedom, which was then used to derive the p-value. Now, in the world of statistics, if your p-value is 0.05 or less, that means your results are significant and it's not happening by chance. For my study, not only was the p-value less than 0.05, it was less than 0.001. So what does this mean? This means that at successfulness, ospreys devoted more time on successful food provisioning, while at the failed nest, ospreys devoted more time on other activities. The difference was strongly significant, suggesting that a nest success is greatly associated with successful foraging and higher rates of food provisioning. Long-term productivity must average 0.95 to 1.30 young per active nest in order to maintain a stable osprey population. For my study, the productivity rate was 0.83, which suggests that the pairs of this study may have had a poor year. On the expansion of forage fisheries, managers can use this data on prey composition, which can then inform ecosystem-based fisheries management. And this would be a paradigm shift from single species, maximum sustainable yield models into a model that considers trophic interactions. American shad was introduced into the Pacific coast from the Atlantic coast in 1871, and since then, they become firmly established. The interactions between American shad and the salmonids are untested hypotheses. We do not know whether they're harmful, invasive species, or if they're just a benign introduction. Perhaps they're an umbrella species, as prey buffers, protecting salmonids as salmonids are commercially and recreationally important. There are a lot of people that I need to thank. Here's my literature cited. For my closing statement, I would like to emphasize the importance of short-term assessments and reports such as this. I understand that my study doesn't have the breadth and scope of a master's or a doctorate student. However, in real-world applications, sometimes managers can't wait 3 more years in order to make urgent decisions. Therefore, studies like this can provide at least a point of reference. Some data is better than no data. I would like to open up the floor now to any of your comments or questions about this study or about ospreys. Thank you.